Alright, enzymes, we already know, are a kind of protein. All enzymes are protein, but not all proteins are enzymes. Enzymes are a specific kind of protein. And we know that proteins are made of amino acids. So we're going to talk a little more about enzymes. An enzyme is a substance that can alter or speed up a chemical reaction without being changed at the end of the reaction. The important part of this definition is that the enzyme is not changed by the reaction. It's the same at the beginning as it is at the end. We call an enzyme a catalyst because it speeds up the chemical reaction. Some places you might have seen enzymes are in detergent. A lot of detergents use enzymes to remove stains. They also use them to clean up oil spills. When oil is spilled in the ocean or in the Gulf, they can dump enzymes on it so that it'll break the oil down into carbon dioxide and water. They also use enzymes in animal feed. Animals like cattle and goats and sheep, the feed that you give them probably has enzymes in it to help it be more digestible to those animals. There are two kinds of enzymes. There are enzymes that break things down into smaller pieces, and there are enzymes that put things together to make something more complex. You already know two reactions similar to this. The reaction of dehydration synthesis is an example of an enzyme putting something together. You know that in dehydration synthesis, two sugars combine to make a starch. Well, that reaction can be made easier or faster by using an enzyme that puts those two sugars together to make it a starch. The opposite of that reaction is hydrolysis. Hydrolysis is the reaction where a starch breaks apart into two sugars, or two or more sugars. And that's an example of a reaction where an enzyme is used to break something that's complex down into things that are simpler. When you name enzymes, generally the name comes from the thing that it reacts with. For instance, in those reactions that we just talked about, dehydration synthesis and hydrolysis, the things that are being catalyzed are carbohydrates. They're starches and sugars. And so the enzyme that's going to work in those reactions is called carbohydrase. You can see that all of the enzymes end in ASE. That's because that is the part of the name that tells us that it's an enzyme. So whenever you see something that ends in ASE, you can be sure that it's an enzyme. There are some vocabulary words that are associated with enzymes that you need to know. The first one is substrate. Substrate is the name for the substance that the enzyme works on. So that's what is either being combined into something larger or being broken down into something smaller. The substrate is the thing that binds to the enzyme to be changed. Enzymes are specific, and that word specific is something that's going to be very important to enzymes. They only work with a certain kind of substrate. Each enzyme has a specific substrate that it wants to bind to, and it won't work with any other substrate. And the product that that enzyme produces is also specific. Each enzyme works with one substrate to produce one kind of product. And the reason that it can only work with one kind of product or one kind of substrate is because of its shape. The shape of the enzyme determines which substrate it works with and which product it produces. The part of the enzyme that the substrate binds to is called the active site of the enzyme. You can think about it kind of like a mouse. It's the part of the enzyme that grabs onto the substrate. That's called the active site. There are four things that can affect the rate at which an enzyme works, and the first one is temperature. When temperature is very low, enzymes don't work well. They're kind of like you. When you get into a hot tub, if it's ice cold, that's not much fun. You don't want to be there. And if it's too hot, then it burns you and it's not comfortable. So just like you have a range of temperature that you feel comfortable at, enzymes have a range of temperatures where they work well. And if the temperature is too low, then they won't work well. If you increase the temperature too much, then it's going to be too hot for the enzyme, and the enzyme will change shape. It's sort of like the enzyme is melting. And when the enzyme changes shape, it can no longer react with the substrate that it was designed to react with, and we say that it is denatured. Denatured just means that the shape of the enzyme has been changed, and so the enzyme can't function the way it was supposed to in the first place. The second thing that can affect the rate of a reaction is the substrate and enzyme concentration. Now you can think about this sort of like an assembly line. If you have an assembly line with people working on it, the people who are working would be sort of like the enzymes, and the materials that are coming down the conveyor belt that they're putting together, those would be sort of like the substrate. So if you have a certain number of people working on a conveyor belt and you add some more people, that's going to make the process go faster. They'll be able to do more. But if you continue to add people until there are just too many people there for anyone to be able to move, they're just shoulder to shoulder and they can't even move, then the reaction is going to start to slow down because there's just no room for them to move and do what they need to do. And so the enzyme concentration is sort of like whether you have a lot or a lot of people or a few people working on a conveyor belt. When there's too many, it doesn't work well, and when there's too few, it doesn't work well. You want to have just the right amount of people working to do the job that needs to be done. Now, the substrate concentration can also affect the rate of a reaction. If there's too little substrate, then there's not going to be enough materials for the enzymes to be able to work on. But if you start adding substrate, then the reaction is going to begin to increase in speed because there's more materials for the enzymes to work on. Now, if you have too many enzymes and it's very crowded, one thing you can do is add more substrate, and that'll help the reaction go faster again. So depending on how much substrate and enzyme you have, the reaction can either go faster or slower. The third thing that can change the speed of a reaction is enzyme inhibition. Now, an enzyme might have a place on it where something can bind, something that would prevent the enzyme from being able to bind to an active, to bind to the substrate. It might prevent the active site from being available, or it might prevent the enzyme from being able to open so that the substrate can bind. Anything that would prevent the enzyme from binding to the substrate is called an inhibitor. And an inhibitor can either be competitive or non-competitive. If an inhibitor is competitive, that means that it binds to the active site. So it's competitive because it's competing for space in the active site of the enzyme. If it is non-competitive, that means that it binds somewhere else on the enzyme besides the active site. And it does not block the active site, but it still prevents the enzyme from being able to bind to the substrate. Anytime an enzyme is inhibited, it can no longer perform the reaction that it was made to do, and so that enzyme activity will either decrease or stop entirely. The fourth thing that can affect the rate of a reaction is pH, and pH refers to how acidic a solution is. On one end of the spectrum of pH we have acids, and on the other end we have bases. And right in the middle we have things that are neutral like water. Enzymes have a preference for their acidity. If they, depending on what kind of enzyme they are, they'll have a certain pH that they like to work at. It's very similar to temperature. There's a certain range of temperatures that enzymes like, and there's a certain range of pHs that enzymes like. Different enzymes have different pHs that they work well at. And if the pH is either way too high or way too low, then the enzyme can actually be denatured. It can change its shape. You can kind of think of it like an acid eating away at the enzyme, and so it can no longer react with the substrate that it was supposed to react with because the shape is different. Alright, now this is a graph that describes the energy level of a reaction either with enzymes or without enzymes. So at the very beginning of the progress of the reaction, we have a certain level of energy. You can think of this as the energy that would be in a piece of food, or maybe bread. Um, and when it's sitting on the table, there's a certain amount of energy available to you through that food. Now, after you eat that food, your body is going to use some of that energy, and that would be the energy of the products down at the end. The very end of the graph down here is how much energy is left over after your body uses the energy from that piece of bread that you've eaten. Now, the change in free energy here is the amount of energy that your body was able to get out of that food. It's the difference between the energy at the beginning and the energy at the end. Now notice that the energy at the beginning is higher than the energy at the end. We call this potential energy. And our potential energy is higher at the beginning because it hasn't been used. The potential energy at the end is lower because it has become kinetic energy. It's helped you move your muscles or do other processes in your cells. Now, in order for that energy to get from reactants to products, we have to overcome this hill here. And the reason that we have a rise in energy before we can get to the products is because the molecules in the reactants are happy the way they are, and they don't really want to change. And it takes some amount of energy to be added before they'll be willing to go downhill and become products. You can kind of think about pushing a wheelbarrow uphill. It's hard to get it up the steep hill, but once you get to the top, it's easy for it just to roll all the way down. Now, that hill is higher for this red line. The red line represents a reaction without an enzyme. And that activation energy, or the amount of energy it takes to get the reactants to proceed to be products, is actually higher than it is when you use an enzyme. The activation energy with an enzyme is much lower. And so basically what's happening here is the enzyme is lowering the activation energy and making it easier for the reaction to proceed to products. Now notice that the amount of energy that you get from the food that you eat, or the change in free energy here, is the same. That energy is the same whether you use enzymes or whether you don't use enzymes. What changes is the activation energy, or the amount of energy needed to get the reaction started. So all an enzyme does is it makes the reaction more favorable. It makes it easier for the reaction to proceed.